I'm really happy, of course, having almost the time. I mean, supporting the research, supporting the student, and supporting a lot of things that we did. Christophe Grova is one of what I call the precious person. So it's very important having those guys behind me because I feel myself more stronger. Christophe is assistant professor at Concordia University, and he is also a member of the epilepsy team at McGill University and adjunct professor at McGill University. He is the head of the team multi Funk LM, Funk. It's difficult for me to say.
Multimodal functional image.
Multifunctional. So this team at Concordia University, but he is also not only a member of the Perform Center, which is now the School of Health of Concordia University, but he is in charge of the physiological suite. So he is the director of the physiological suite of the Perform Center. So of course I know Christophe for a long time, and he did his he started his research program in PET and inverse problem in PET. He did, I mean, a lot of input in this area and moved to EEG, fMRI, and we understood his evolution over time. So he becomes like the multimodal imaging guy that I know over the world. So he has a lot of expertise in different modalities and how we can combine between all of them. So I'm really happy that Christophe is still supporting, I mean, us and supporting the research and supporting the student. So he is going to present today an overview about the EEG and MEG and source reconstruction from the dipole modeling to advanced imaging of brain networks and functional connectivity. This is his first part, and this afternoon I am going to introduce him for the second part of the research program. So thank you very much, Christophe, for all your support and your help.
Okay. Thank you. Good morning, everyone. Thank you, Habib, for the nice introduction. And it's my pleasure to be here to this very nice workshop. So I'm going to talk to you about EEG and MEG and source imaging. I had planned something a little bit more extended. I'm going to cut some parts because I am aware that I am largely overlapping over the lunch break. So to try to keep you awake, even if I can show you what is the sleep EEG rhythm, but we'll try to avoid that. So, so this is my presentation. So I had few slides on the origin of EEG and MEG signals that I guess because what I see from the audience here, I'm gonna skip them very rapidly. I will just perhaps show that one because that one is kind of interesting and linked to what we are using, especially in my work in epilepsy. So here, this is a scalp EEG signal. So for those who don't know, you have roughly 60 electrodes. Between two bars, you have 1 second. And this is an epileptic patient. So this is not a seizure, but these little elements here are what we call interictal spike. So these are short transient discharge that are typical of the epilepsy of the patients. So we can localize them, and that's one of the core of my research is to find out the brain regions that are generating those. And it's not a seizure, so the patient is not moving, so it gives us a good idea to map what's going on. The only reason I'm showing you this here is to illustrate the inverse problem, is that these are very interesting data, and we are actually planning to acquire our own data now. So these are scalp EEG, and together with scalp EEG in that study, they record intracranial EEG. This was a grid, the NICOG data. And so here you see that at the time of what you see here on the scalp, which are few spikes, there is way more going on inside. Okay, and that's just to show you where we start from. So we try to, in a way, localize that from what we have from the scalp, but we really do see only the tip of the iceberg. And that's going to be one of the message. And the other message, to see this epileptic discharge from the scalp, we need a relatively large spatial extent of cortex to be activated. And that's going to be one of the methods I have been working with Jean-Marc Lina, with Habib, that I'm going to show you later. You heard a bit about that method with Jawata before. So I will skip some of the basic recording, what is EEG and what is MEG. So just that you are cortical. So I will just skip those, because just by interest of time. And let's start with, so if you have questions on those, I can of course answer them, but I'd like to start with, we are recording scalp signal. Those scalp signals are mainly generated by synchronous neuronal activity that create a signal on the surface. And we have two main techniques to measure them on the scalp. Either we put EEG, so we are measuring the electrical field, or because any current in a dipole, as you know from your physics class, any current in any movement of charges in a wire is generating a magnetic field. So your neurons are generating magnetic field that we can record using MEG magnetoencephalography. But at the end, it's the same idea. And now I'm going to mainly stay concentrated first on how we can go from scalp and go back to the brain generator, starting from the very early technique to what is most advanced. And in the second part of the talk, I will show you how we can use that to try to map interactions between brain regions to talk about network activity. So I'd like to start with that one. Some of you know that very well. It's a nice little sketch from Mattia Malainen, but the inverse problem looks a little bit like that. So what you're looking for is like one day on Earth, there were dinosaurs, and then they moved through what I would call in EEG the forward problem. So this is what remains nowadays. And from this, us as investigator, trying to find piece of evidence. So we are trying to shake the data and find piece of element, as these scientists are doing here, finding bones, finding piece of that. And then what we are doing in that inverse problem is to put all those pieces together to come up with a model of what was at the origin of that, which are, in our context, the source of our signal in the brain. And more likely, we're all doing something like that. Okay, so it's not bad. It looks like a dinosaur, but we are making mistakes. And I want you to be aware that solving this impulse inverse problem, we might do that most of the time. So let's go back to our signal. So as I told you, we have generators mainly on the cortex, because we know that the signal intensity is decreasing 1 over the square of the distance. We are mainly, I don't go into detail, but we are mainly talking about postsynaptic potentials. And if you have signals, we'll go there. If you have signal on the cortex here that is synchronous, you're going to have a dipole, current dipole, that's going to generate an electrical potential, and it's going to generate a magnetic field that you can record. So you have, for instance, if you have a synapse here, you have motion of charges. So if you have motion of charges like that, it's going to create a magnetic field. And because you don't have accumulation of charges and around your neurons, it's a liquid, it's a conducting system. So some ions will close the circuit, and this volume conduction current will create your EEG. So let's go from a bit of modeling. If I have a dipolar source here, in terms of EEG, that would be the electric potentials associated to this current dipoles. Positive here, negative, and it's 0 when you are orthogonal to it. And you place that on the real EEG measure on the scalp, you get measurements like that. Then we can do the same for MEG, which is we have our dipole, but this time it's going to generate a magnetic field, so rotating around. So we'll have ingoing and outgoing magnetic field rotating around. And if I place a helmet of 300 MEG sensors, this is what I'm doing. So this is actually an actual recording one of the very first we recorded not far from here at the MEG at University of Montreal, like, yeah, almost 20 years ago now. And actually it's very interesting because you see directly the electrical and the magnetic activity at the same time. And this is very interesting. And now, of course, we have different types of generators. What we know in EEG is that we are mainly sensitive to those radial currents that are generating a very large field like that. Whereas in MEG, in theory, we're not that much sensitive to those. We are more sensitive to those which are a little bit more discrete in EEG, but will give us a strong signal in MEG. And so the two are very complementary. So I'll start with the basic equations, but with some drawings. So the idea is we are looking, so this is our main generative equation. So we have some signals that we are measuring from the scalp using n electrodes, typically around a few hundreds, like from 60 to a few hundreds, 200 or 300. And the idea is we want to find out our source. Our source are rarely a single point, are more distributed as a network on the brain. And then we know by solving actually the physics of the problem and Maxwell equations under some conditions that if I know where is the source in the brain, I can predict what's going to be the signal on the scalp. And this is what we call the forward problem. This is the matrix G here. So if I know where is or the lead field, so I know how to make the link from the brain to the sensor. And now our problem is to do this inversion. Of course, we need to take into account noise. And now the problem is to go from the signal back to the source. And of course, as I say, this is a heel-post inverse problem. So that means that this problem has an infinite number of solutions. And I need to make some choice to find one particular solution. And I will go over some of the main technique. I'll show you the very first technique that has been proposed in this domain, which is assuming this has been generated by a single dipole. And you can localize a single dipole generating that, or more imaging technique, which are assuming what we call distributed sources. So we are assuming that the generator mimicking what's going on in reality are the pyramidal cells oriented along the cortex, and those pyramidal cells are all perpendicular to the cortical surface. So if I put a lot of dipole perpendicular to the cortical surface, at each time sample, I can estimate an intensity. And this is what we call source imaging. We have an image every millisecond. Here we have a point every millisecond. Of course, I need to talk a little bit about inverse and forward problems. So inverse is we start from the data from the scalp and we want to find the source in the brain. And this is the difficult problem. It's ill-post. It has an infinite number of solutions. And we have to solve the forward problem. And the forward problem is a modeling problem, which means if I put a source somewhere in the brain, if I'm solving Maxwell equation, I can predict what's going to be the signal from the surface. Either this would be the signal in free space. This is direct. We know exactly what it is. Or the signal after it has propagated through the different layers, including the skull and the skin and so on. And then you get a signal like that. So source localization in a nutshell is just an optimization technique. Is like you place a dipole, you apply your Fourier model. So this is a computed for field, a computed tomography. You compare with the one you measure, and you do the optimization until you find a solution. You will always find a solution. The problem is you can find more than one solution. You need to deal with the Fourier model. So EEG source localization had bad reputation in the old days because people were assuming the brain or your head to be a sphere and the skull to be a sphere. And this is a very bad model for EEG. And nowadays, we are able to do way more accurate model using boundary element model, using the different layers like that, modeling the effect of the skull. So now we can do very good job in terms of EEG source imaging as long as we model well the anatomy and also the effect of the skull, which is important. And for a long time, it was easier to do source reconstruction in MEG because MEG is not that much sensitive to the skull. So assuming a spherical model for MEG was working better than for EEG. And so now we can have overlapping sphere models or boundary element model, but with one layer, assuming that there is no generator after, no influence after the envelope of the brain. And of course, I'm not talking about that here, but to do a proper job, we need to handle the geometry properly. So we need to know where are our sensors and usually where is our brain and our head model. So in an ideal solution, you have a digitized version of your sensor, you co-register with the actual MRI of your subject, and you have the actual anatomy of your subject together with the scalp sensors. Of course, you can also do things when you don't have that playing with templates, but you're going to decrease the accuracy. So the best case scenario is when you have that, and also make sure you do the co-register properly, because lots of systems are just finding the position of your electrons by using 3 anatomical landmarks here. So placing an nasions and 2 periauricular points in 2 data sets might be very noisy in terms of the accuracy of your registration, and we have ways to do that by just sampling more points on the actual head of the subject. So let's go now for source reconstruction technique. So I'll start with the earliest one, which was implemented in a very famous software in our field called BESA, which is still a very good software. And this was proposed by, there were dipole fitting before that, but it became very popular by Michael Shirk in the 80s. And this is the idea of the time-varying equivalent. So the idea is you have a generator of brain activity, which is supposed to be a few square centimeter of cortex, and it's going to be modeled, this little patch is going to be modeled as one dipole. And you will fix the position of the dipole, and then you allow the dipole to change in amplitude, but also to change in orientation. And then you try to fit that with your data. So usually you need to have relatively good SNR data, and you're going to solve the problem because like each dipole is 3 position, 3 orientation, 1 amplitude, so 7 parameter. So if I place 3, 4, 5 dipoles, I reduce largely the number of unknowns compared to my 100 of sensors. So I have a problem that becomes better condition that I can solve by linear technique. It's non-linear in terms of the position, it's linear in terms of orientation and amplitude. So in the problems of in the setup of Michael Shirk, the dipoles are fixed. He is finding the position first, and then is fixing them with time, and then finding the amplitude along time. And there are laws to vary. So the typical problem would be like that. You get a signal like that. You want to explain a signal like that by 2 dipoles here. So here you have a radial dipole with its topography, a tangential dipole with its topography. So let's assume you find out that you have 2 sources in your signal. Then it's a very basic linear model that's going to find out the time course associated to that in a least square optimization so that you have only noise remaining. So this is to find the amplitude, and you have the time course. And what is very cool about that technique, all those electrophysiology technique, I was fast at the beginning, is the time resolution, of course. We are at the millisecond scale, so we can clearly measure causality. We can clearly measure that the activity is propagating from a region A to a region B, which is not clearly not the case when you do fMRI and other techniques. One thing to know is when you submit, well, as any model, but I'm saying that because most of the software are using that. So it's a linear fitting model. So you explain part of the variance, and most of the software are showing you the residual variance, which is 1 minus that would be the goodness of fit, which is the variance that the variance not explained by the model. So your model is a source here, an orientation, and then you apply the forward model. So this is explained by your model versus your data compared to the variance of your data. So this is a value between 0 and 1 that gives you how much of the data was non-explained. So you want this to be very small, or you want 1 minus that, the goodness of it, to be as close as possible to 1. And I'm just showing you that because this is a metric that I still see that a lot in many papers, that people will say, I did a good dipole localization because I have a very low residual variance. Okay, you need that, but it's not because you have that you have a good localization. I'll show you a few examples. So we'll do a dipole localization for fun using a toy example. So let's assume we are in the context of MEG. So I draw a magnetic field rotating around my true dipole. So let's assume the true source is this one, the green one. And then I want to fit that. So I'm just getting a signal like that. And then from that signal like that, I want to localize that. So all the dipole fitting algorithms are iterative. So what they do, they place a dipole randomly. They compute the forward model. So you get the field, which is computed here in dotted line. And they measure the correspondence, which is this dotted line and that one. And you want them to be as close as possible. Of course, here, they are very far from each other. I'm almost explaining nothing. I have a residual variance of 90%. And then you let the algorithm go. It's going to optimize. Oh, 30%, it gets better. 10%. So now we are close. And now we know that in positions we are quite close, but we have other degrees of freedom we can play with, which is the orientation. So I can change the orientation. I get 1%. And finally, I do the linear model I showed you before. And you fit the amplitude, and you get this. So it's not exactly. You need to handle the noise. We have a 0.1% residual variance. It means that we explained 99.9. It's really a lot. And of course, you might say, OK, why not? Of course, in linear model, people are well aware of that. But in the field of dipoles, when they play with their software, sometimes they want to go even better and to try to add more dipoles. And what's happening if you add one more dipoles? It's decreasing, and two more, it's decreasing even more. Of course, as long as you start adding more dipoles, your residual variance will decrease, but they don't explain anything because you clearly are doing overfitting there. So that's very basic in modeling, but people, when they are using the clinical software, they might just use that and still continue. And I will just give you a practical example. And this is a very good book if you want to read about the basics of doing source imaging in MEG, notably. It's by Rita Salmelin. So this is a median nerve stimulation. So you send a median nerve electrical pulse here, and then you get a somatosensory response, few milliseconds. So you see that you do that several times, you average your response, and you get a first response with this topography 20 milliseconds after, then it goes like that, and then a few milliseconds later, it goes down on both sides. So if I start here, it starts there, go a bit posterior, and then down on both sides. So you see, it's a very dynamic process. And people who know well how to use dipole modeling can actually get this very accurately. And to do that, you need to fit 4 dipoles here. So you have your first dipole localized here that's getting the earliest part at 20 milliseconds. Then the second one a bit more posterior with the parietal regions. And then 2 late ones here and here. So here we are in the primary somatosensory area. And then we are in the secondary somatosensory area, contralateral and ipsilateral. So the activity starts contralateral, and then it's also ipsilateral, few milliseconds. So this is actually, and the residual variance, or the goodness of it, was above 80%. It's a very good fit here. And this is a very nice summary of what's going on, because with one picture like that, what you get is where is your activity localized, and what is the time course of those propagation. It starts there, spread there, goes there, which sometimes is not that easy when we are doing full imaging and movies and complicated movies. So it's a very nice summary, but it's not always that easy to get. And I really challenge you, if I give you those data and did not give you any prior information, to guess that you should use 4 dipoles to do that kind of localization. You might get the first one, but you will probably never get that. So that's why it's almost an art to do good dipole localizations, because you need good knowledge of the underlying physiology to guide how many dipoles. So you place your first dipole, you fix the position, you add a second one, and so on. So in complex cases, it's getting more and more complicated. And also, what's more complicated, it's how many of those dipoles I should use. So this is another example with an interesting study in epilepsy done by Humitaidin before he started his postdoc with me. And here they looked at clusters of dipole from the very early version of an epileptic spike to the peak of the spike. And you see a very nice propagation from the medial structure, from the hippocampus to the lateral regions. What you have to remember here is that we are modeling point-like activity. But we know from the beginning that the activity in the brain is not one point that is activity. It's probably an extended patch. And I'll show you one example, typically in temporal lobe epilepsy, which is the standard example in epilepsy. This is a MEG spike topography. This is a very huge signal on the scalp from a temporal lobe epilepsy patient. And if you localize that with a dipole, you get your dipole, very nice fit, right in the hippocampus. And this is a wrong localization. This is a standard error. Why it's a wrong localization? Doctors will like this localization a lot because we know the epilepsy is starting from the hippocampus. So it's a good assumption. But that localization is wrong because to get such a strong signal, I showed you at the beginning, you need to have something spatially extended. So you need few square centimeter of cortex to be activated. And now you try to explain a generator of few square centimeter by one point. So at best, you would get your point at the middle of your patch, but it's most of the case, you get a deeper dipole that when you project the activity, when you spread the activity, when it's going to be the spatial extent on the cortex. It's like when you have a flashlight. If you put the flashlight inside, then you go a little bit later, it's larger and larger and larger. So that's a little bit of that. Whereas here, the right localization would have probably been something extended. I don't say that there is nothing inside. There's probably something inside also, but it has been a mismodeling here. So what can we do next? Another technique, very famous technique that has been proposed to go a little bit beyond this idea of fitting dipoles and also to answer the question, how many dipoles should I use, has been what we call spatial filtering technique and notably beamformer. So the idea here is we're going to place a grid and so I'm going to place a grid, and for every point of the grid, I will try to map a quantity, which is the likelihood to have a dipole there. And in spatial filtering, what we use in beamformer, we use a spatial filter, which means as in the time domain, sometimes you want to do bandpass filter to filter between two frequency bands. Here it's spatial filtering. So I want to build a filter, which is a matrix that I'm going to multiply my sensor, which is what you see. This is the spatial filter here. And the objective of this spatial filter is to extract the signal from this source and to switch off the signal from all the other sources. It's a little bit like it's coming from radar. So it's looking at what's coming on from that particular and erasing all the rest. Of course, you cannot erase completely, but you try to reduce the variance. And by doing that, for every point, you have a spatial filter, and you can create a time course. So if I have my signals, I create a spatial filter for that point, and I have my signals. So now I'm going into the brain, but I'm not solving the whole imaging problem. I'm localizing point by point on the grid. And then I can do lots of fancy stuff. Like, for instance, if I have a control and an active window, I could have the power in those two windows and make a pseudo Z map of the activity in a specific mode. And that has been used a lot for motor response. So this is when you do the movement of a finger, which is not an easy task to localize with a single dipole. So here you have a map, because here we're going to take into account that when I'm doing that, there are specific oscillations in your brain that occur. Notably, you have some mu rhythm, which is close to the alpha band, but on the motor cortex in the preparation. When I'm getting prepared to do the motor activity, I have increase of mu rhythm in the motor cortex that has been localized here. And then after the motion, you have what we call a rebound of beta activity. So right after the motion, which is here, so you have the decrease in alpha here, and you have the synchronization in beta just after. And beamformer are particularly good technique to localize in those specific frequency bands, and that works a lot. And what beamformer has been very useful as well is for denoising. It's a spatial filter, so it's trying to localize what's going on here and removing all the effect. Sometimes if data are correlated, typically in auditory conditions, if I have two purely correlated regions, it might get into problems, but in practice, it's rarely the case. But this is, to give you an example, this is presurgical mapping. These are kids from Toronto. This is the team from the Sick Kids in Toronto, from Duke Chain. So here they were doing auditory mapping. So they do, here it's a healthy subject. They do a standard auditory mapping, just hearing beep, beep, beep. You get the average, and then you get your localized after beamformer. So here, the beamformer is a grid, is like an image, and then the good way to do it is to take the maximum, the local maximum on that, which are the two dipole localization. So a very clear auditory localization here. But then they wanted to do the same on another kid who had dental braces. And of course, you have lots of kids who have dental braces, and MEG is not the best tool to do that because MEG is highly sensitive to magnetic artifact. So if you do that, this is the signal that you get. So kids with dental braces, your MEG signal is completely, because those signals coming from metal in the MEG is a very sensitive design, are gigantic. The cool thing is, spatially, they are localized. Those artifacts are localized here. Our spatial filter is working here. So this spatial filter is extremely efficient to remove the effect of those. And that's what you see here. This is the average potential. You don't see anything, but the localization was still very good. So this is for beamformer. That's another example of beamformer activity here in epilepsy, where people look during a spike at a specific frequency of interest. This is the group from Lyon with Julien Jung. And what they did, you have the scalp topography, and they did a map, a beamformer map in a specific frequency band. And then they try to find out if you can change the threshold of that map to get a notion of spatial extent. I think they pushed a little bit too much where the beamformer is used, because again, the beamformer is not an image, is fitting a dipole one after the other. It looks like the spatial extent of what they get is not so bad, and it seems useful, and they published that in Brain, but it is still very sensitive to the noise level, because you are doing a dipole fit for an extended sources. So the last kind of technique I wanted to introduce, which is really what we call source imaging technique, is the distributed models. So we went from dipole to dipole scanning. We place a grid, and I place a dipole at each point, and I'm trying to get a metric that says how good is my dipole. Beamforming is the most famous, but there is also music scan, and it's different version. You have rap music, pop music, and etc. And distributed models. So distributed models, what we do is we start by linearizing the problem. I told you the problem is non-linear in terms of position, it's linear in terms of amplitude. In terms of position, when you change the position, the effect of the signal is 1 over the square of the distance, so it's non-linear. So what we're gonna do now, and it's based on what we know about the anatomy, so this is a real view of the cortex, and you see that our main generator, which are the pyramidal cell, are oriented like that, and they are all oriented, they tend to synchronize a lot, and they are all oriented parallel to each other and perpendicular to the cortex. So what we do with more advanced neuroimaging technique now, we use anatomical MRI, we extract the cortical surface, and we are distributed dipoles all along this. So for each dipoles, I know the forward model. So now my object to localize looks like that, where J is not few time course of 4 or 5 dipoles. Now it is 8, 10, 15,000 dipoles distributed all along the cortex. And now what you want to do, if you fix the orientation, it's at every millisecond, you want to find the amplitude. So this is what we call source imaging, because reconstructing J, it's really a full, it's along the cortical surface, but it's a full reconstruction of the whole surface at every millisecond. So you can get a movie. So the good point of that is that the problem is becoming linear, but it's still underdetermined, because typically we have M, which are our data by the order of 1 or 200 sensors, and we have several thousand of generators, typically 8 to 10,000, and a lead field matrix in between. We need to handle noise, but we know how to solve that mathematically, like using optimization, or more interestingly, using the Bayesian framework. I will not go in all the details, but if you put assumption on this underdetermined linear problem, you can come to a solution. But it's the same idea. And in the dipole model, I was making assumption that I would like to find a source with 1, 2, or 3 dipole, and I will find my generator. Here, I need to put an assumption here for my regularization, and depending on the kind of assumptions, or if I am in the context of Bayesian framework, the kind of prior that I put, I'm going to get a solution. So another method is another prior, and I will go through a few of them. So just again, so we have the hill post. So there are, I will go over the main, the most famous one. So the main one, which is quite famous in mathematics, it's called Tikhonov regularization, is the minimum energy solution. So basically the problem looks like that. So I want to find a distributed sources J, so that Gj is close to M. So I want a good data fit. And among all the possible solutions, I will choose one where the amplitude of J, the energy of J, so J square, is small. And I can also weight that. So I can weight more if you are deeper or not, or on the cortical surface. I can have fMRI weighted minimum norm. So if I put some weight from fMRI in W, and all kind of methods there. Well, this is the standard way. There are very elegant way to show that within the Bayesian framework. I will not go there, but this is really the prior distribution on J. You define a prior distribution on J with a source covariance model, which in the case of minimum norm is the identity matrix, or a weighted version of that matrix. So this is giving you one solution. You have other technique. Around the same time, the technique Loretta was proposed, which is quite similar. And the idea here was to minimize the Laplacian of J under the constraint M equal Gj, which actually is equivalent of using this, but putting the Laplacian or half of the Laplacian in W here. What is the Laplacian doing? We're talking about a spatial Laplacian. So this is imposing spatial smoothness. So if you have two dipoles close by, you don't want the activity to jump from one to the other. You want a solution that is smooth spatially, which along the cortical surface, I think that makes sense. But when people were doing that at the very beginning, they were using very crude grid every centimeter. So sometimes you are imposing strong smoothness between 2 points here in 2 different hemispheres, which might not be so on. But that technique was proposed. And I'll show you the method that we propose. Jawata showed that a bit this morning, the maximum entropy on the mean. So just to show where this started from, these are some of the earliest publications on minimum norm or on Loretta, both in '94. As you can see, was perhaps not so exciting as new imaging technique. So you get a map. The head model was a sphere. You get a map in the sphere, but between that and a sphere with a dipole, there was not so much difference. Now we can get way better, and now we get actual model from MRI of the tessellated surface, more accurate forward model. I'm not talking about that, but it's very important to model the noise covariance matrix. And you have also a way to represent your result on the cortex and to inflate the cortex to see a little bit more what's going on in the gyrus and to compute statistics. So this is the way modern minimum norm is done nowadays within most of the package, Brainstorm being one of those. And now you can even do that on resting state or single trial data So it's a projector. You project your data on the cortical surface, and then you can do all your statistics there to make very nice data. This is one study. I think this was using S. Loretta, which is a normalized version of minimum norm during spike when you're using like during a propagation of the spike from the earliest phase to the peak here, and you see again some activity starting deep and trying to go a little bit more on the surface. And that was an interesting study. They were either using MEG only, EEG only, or MEG and EEG together within a fusion paradigm. The main question here, again, how can we threshold those maps? And we have shown in many papers that the extent of those maps is highly sensitive to the noise levels. So we wanted to go, and that's more specific to my work now, in the context of estimating the spatial extent of those generators. And that's where, so Habib was also at the origin of that, and with my colleague Jean-Marc Lina, we proposed the maximum entropy on the beam as a technique to do so. And why spatial extent? First, because what we know from the literature is to see an epileptic discharge, which is a strong ISNR signal on the surface, you need few square centimeters of cortex to be active. Typically, in EEG, we believe it's 4 to 8 square centimeters. MEG is more sensitive, so perhaps 3 to 4. And those measures were done using simultaneous recording with scalp and then grid. But what you have to remember is also the grid is just seeing a part of the cortex, because there's lots of regions we don't see with grid. So this estimation is probably an underestimation. And there are other problems with grid, is the fact that when you place a grid, which is a plastic, like you have the plastic between the electrodes on top, then you're going to have attenuation in the middle and amplifications where you have the hole around it. So it's not that easy, but there is a special extent. So this is where we came with this method. I have just 2 slides on the methodology, and then we'll try to go on more into the applications of a method that I think we demonstrated that is sensitive to the spatial extent. So we have the version in the time domain and the version in the time frequency domain using wavelet extension. So the basic idea, it is a Bayesian technique. So we start with a prior, with a model, and then we have the set of all the distributions that explain the data. And we're going to make the measure of the difference of the distance between a solution that explains the data and our prior. We're going to use relative entropy or Kullback-Leibler divergence to measure this. And then among all the solutions that explain the data, we're going to use the closest to our prior in terms of a relative entropy. So this is actually very, like, not far from maximum likelihood in the Bayesian domain. It's just we are using entropic technique here. The cool thing of using entropic technique is we have a little bit flexibility in our prior model. So we're not playing only with the noise covariance. Actually, we are using a model like that. And perhaps Jawad explain you a bit about that, which is assuming brain activity to be organized as parcels. And each parcel is controlled by a hidden state variable. So each parcel can be activated or not. And if the parcel is not activated, we have a direct distribution, so it's switching off. If it is activated, I have a Gaussian distribution, so a kind of minimum norm prior, or a bit of low beta. And we show that this is working quite well. So this is just to say, actually, if you want to see, this is the main paper, and Cécile Ambla was with Habib working on PET on that technique. This is the main paper showing you the proof of that. And the interest of that, I won't go into detail, but it's been also mainly developed here, locally, physically here, is that you can solve this problem, and you can show that solving this problem aims at solving this equation here, which is a nonlinear optimization, but in a space of dimension, the number of sensors. So you go from a space of 8,000 dipoles to 100 sensors. So, and you can show that this function is convex. So you have a solution for that you know. And what's good about that is now everywhere in the metro, you can see that everybody is talking about NEM, and we're happy about that. So this is for the Musée des Mémoires Montréalaises, but
I'm happy to see MEM everywhere in the city. So, application of it. So first, we localize spikes. So just to give you an example, this is minimum norm of an epileptic spike, and this is MEM. MEM has a quite nice spatial profile, and usually the doctors working with us like the profile, so we have something like that. We carefully demonstrated, so Giovanni Pellegrino showed that it's an efficient method. In this first study on 49 patients, we compared MEM and dipole fitting, and compared to the ground truth, so the known localized epileptic focus, we get a good concordance in 80% as opposed to 70% for the dipoles. But dipoles was already working quite well for a very basic method. And then we apply distributed models, and this is just to show you on the same spike here. This is our MEM technique, non-thresholded compared to minimum norm, S-Loretta and ESPM, which are two normalization version of MNE. And so the maximum is well localized, all that, but the spatial spread, which depends highly on the noise level, is way better estimated with MEM. This is what you see, distance to the focus, and the spatial spread, MEM is better. And you have a bunch of other results here. So we still have the problem of thresholding those maps, but what is nice with MEM is that we have a strong contrast because of this ability of the parcels to switch off. And usually we have a very nice contrast, so clinicians can even use our map without thresholding and get relevant clinical information. I will not show you that now, I'll show you that this afternoon, but we carefully demonstrated that this is accurate. And I will just go a little bit about few oscillations, and I'll switch to connectivity. Why do I want to localize oscillations? Because for now we localize strong SNR data localized in time, but sometimes you want to localize oscillatory activity, and notably when we are looking at resting state ongoing activity, that's exactly what we wanted. So this is the wavelet-based MEM. So the principle is not very complex. So we use discrete wavelets. I will not go into the details, but discrete wavelets have very nice properties on your signal. So it means you summarize your signal here by a non-redundant plane here with different frequency or scales and time. And so you have exactly the same amount of information. And then if you have a box here over all the channels, you have something of the dimension, the number of channels, you can apply MEM to reconstruct that box. So we are reconstructing in the source space, the time frequency domain, and then I can exploit it to look at oscillation in a specific band to reconstruct everything. And that's what we did in this example on an epileptic seizure. So localizing an epileptic seizure, what you see is that so this is MEG, this is EEG, and this is the beginning of the seizure where you see ongoing rhythmic activity. And localizing that in time is a kind of very low SNR signal. So what we did is we look at the onset, we applied standard time frequency analysis. So we saw, okay, the frequency of interest were around that. And then we look at the discrete wavelet. So we localize where we found interest. And then once we define where we want, we can apply on all those boxes WNEM. So if I look exactly at my onset, I know I'm interested here in the high beta band or low gamma, so 18 to 35 hertz for that specific example. So I look those, I make an SVD of those, and I get a map that we said was a map of the seizure onset. Just to give you an example, we had a patient who had a seizure here. So you see a low amplitude seizure, starting to be rhythmic like that. And actually the same patient had several times the same seizure. And that was very cool for us because then he has a dysplasia, he has an abnormality that is localized here from the MRI. This is standard spike localization. And now we use this technique wavelet, and we find with the technique of the wavelet, we find for all the seizure a very concordant localization. And we can use that in other contexts. This is another study when we look sometimes around an epileptic spike or just before, you have a fast oscillation. Sometimes we call it that it can go very fast sometimes. So here we were from 40 to 160 hertz. Sometimes it can even go above 200 hertz, which is what we call fast ripple. And in this case, this was identity EEG we find very good concordance when we localize this fast oscillation using wavelet MEM and standard spike using standard MEM. And what can we do now when we have nothing, when we analyze resting state data? And can we still do something? And so I'll skip that because that's exactly what Jawata showed you this morning. So Jawata showed you that you can do that in comparison with the SEG Atlas. We'll skip those slides. So it's actually working quite well. This is another study where now people are also trying to localize resting state activity even in patients in different bands when there is no epileptic activity, and when compared to a population of subjects, Z-scoring it, normalizing it, they found that patients are showing ongoing abnormalities. So even during resting state, their oscillation in the beta band, in the theta bands are not normal, and you can use that to create abnormality map as they did here, and that could predict surgical outcome, as they were showing here. So our take-home messages for source reconstruction. So several hypotheses are associated to each localization method. So when you choose a localization method, you need to know what are the assumptions you took. So we can start from very basic dipole fitting, which usually requires good knowledge of the physiology, to advanced source imaging. We have less parameter to tune, but we get way more data, and interpretation is a bit more difficult. All methods are useful. Remember my slide from the beginning. Some might be wrong, so that's why it's always good to generate the data a bit differently, like you do different averaging, different bootstrap, and you localize not once. You need to do statistics. So a lot of people are doing one localization. It's better to do several localizations and to look and to compare the different methods. So it's always a plus. And for spatial extent, we mentioned that MEM is working well. It's available in Grace Thompson. So I will now switch to the second part of the talk when I'm going to talk about connectivity metric. And as written here, I got mainly those slides courtesy from my friend, colleague, Dr. Karim Gerbi from EDM. So I'll go over those slides and I have a few more examples, but do I have questions before we go there?
One question regarding the learning data. So I have the limitation of the data we want to apply to EEG. Is, for example, number of lenses that we have, is it a limitation, or is it we can just apply it to
So the question was about using beamformer and the number of electrodes.
Yes.
Number of electrodes is a question for all sorts of reconstruction technique. So I would say that around 40 electrodes, we start having good results. We recently published, not with beamformer, but with other technique, that you can also have, when you know what you localize, typically an spike, you can have also non-regular montage. For instance, you can use a lower 10-20 system, so 20 electrodes, and locally where you are interested, you put a little bit more density, and you can get very good results. That would be a cheap way to get decent results for source reconstruction. And we just published that a few months ago. And for beamformer, it's working also. It has the same sensitivity. I would say 40 to 60 electrodes is better. 20 is feasible. It's going to be a bit more blur. The main issue with EEG compared to MEG is beamformer is highly sensitive to your forward model and to your noise components. You need to pay real attention to those two items. And that's why it's been used more for MEG than for EEG, because the forward model is a little bit easier to solve for MEG. So if you pay well attention to that, you can do this job. And other question? Okay. So I'll switch to the slide of Karim. And so now I would like to go about the notion of measuring connections. And measuring connections, what we're going to see is we are lucky in EEG and MEG that we have a very a method that are very accurate in time, very good temporal resolution. So this allowed us to make it's very rich in terms of the information we can get from that, in terms of the notion of coupling. And as opposed to people who have used fMRI, for instance, in fMRI, when you look at connectivity, a great, not only, but the great majority of the people are using only correlation. So synchronicity. So what is synchronicity? We'll start with this toy example by Wiggins. And this is a very famous example where you have two pendulums. And if you put your two pendulums like that, they're going to be completely random. But if you place a link, here was a wooden link between those two, so the vibration of one would transfer to the vibration of the other, as it's done in this experiment. They will try to synchronize each one, because you have two oscillators that's going to start being linked. And this is what kind of measure we are measuring. And we are different kind of relationship. So we'll see if we are measuring generators or oscillators that are in phase, anti-phase. They can be a synchronization but with a fixed phase. The phase is not the same, but the phase difference is the same, or something completely random. And then the idea is that we are measuring something like that when you start listening to the noise you have in the stadium, and that's exactly what we do when we are measuring brain activity using our EEG and MEG signals. All our neurons are active. You will never hear what this specific guy is saying, and you will never hear in EEG or MEG what this specific neuron is saying, but you hear a global noise. And when they are starting singing or making a rola or things like that, then you start understanding why, because they are all in synchrony saying the same thing, and you start understanding. And you can use more than one channel to measure. And this is exactly the kind of recording we have in EEG and MEG. And you can not tell what an individual is doing, but you can measure the synchronization over that crowd. And hopefully, when you have more than one sensor, you could localize them. You can say, okay, this synchronization of this song is coming more from that corner than from that corner. That's what we're going to try to do. And where is that coming from? You can have the analogy that it's an orchestra, but it's not an orchestra with one conductor. You don't have one chief conductor. It's more self-organization, so it's more something we would see, for instance, in jam session, in jazz, or something like that. So that's another analogy. So what I will go again, I will probably not do everything, but let's look at some tools that have been used and give you some example, and also some of the pitfalls that what can be done and what cannot be done. I'll try to cover that in 15 minutes. So the first thing is correlation. So this is the most famous way. So you measure the cross correlation, you normalize by the value. So you have a value between -1 and 1. Very standard metric. Probably the main one that is used to measure connection in fMRI. But the problem here is that, especially if you have one main oscillations in your signal, this measure will be mainly biased by that oscillation. So you're not getting that much information, and it's also mixing information in frequency, so the kind of oscillations and the information you would like to have two types of measure when you have generators. You want to see what's the link between amplitude and what's the link between the phase. And this is not the ideal technique. So usually we prefer to go for coherence. And coherence is a kind of cross correlation, but in the frequency domain. So and the cool thing is you can compute it at every frequency. So you can look at it in the alpha band, in the beta band, and so on. And the basic idea is you have your power spectrum, X and Y, and you have your cross power spectrum, PXY. And then you can have one measure. Here we take the modulus, because these are complex numbers. So if I take the square, I get just one value, which range between 0 and 1, and which can inform me about the coherence. So how much link there is between my two signals at a specific frequency. So I facilitate that in the frequency domain. What is even more interesting in this case, we talk about coherency. So in terms of coherency, what we did is we just removed the modulus. So now here you have the cross spectrum here, but this is a complex number. So you have two main informations here, because you have a real and an imaginary part. So you have an information about the link in terms of amplitude between your two signals and the link between the phase of your two signals. And it's very interesting to decouple those two because these might be very different mechanisms underlying. So if two channels are coupling, they are more likely to show a consistent phase difference. And one technique to measure the instantaneous phase is the main one that has been used is Hilbert transform. You have other technique. You can use wavelet. And there are techniques that has been measured to measure phase consistency. Okay. And one of the techniques that is quite famous, the first one, is what we call the phase locking value. The phase locking value is this one. You take typically Hilbert. So Hilbert is decomposing your signal into an amplitude, an envelope, and a phase, an instantaneous phase. And then you have the phase of your channel 1, the phase of your channel 2, and then you can make the difference between those two phases. And then along time, you can measure it. And if the difference is, like in the first example, when they are in phase, so either the difference, if they are purely in phase, the difference is 0. So they are really all along here. Or the difference might be non-zero, but might be at a specific value. And if you average several values, you get one value that gives you how much phase locking you have in your signal. And usually you'd like to make a statistics on that. So what you can do is you can shuffle your second data and you can make surrogate data to see how much of your coupling is getting out of your noise. This is a very interesting technique. It is non-sensitive to zero lag correlation, and I'll show that in a minute. So what are the important interpretations of that? We are measuring connections, and you have approximately the same issue with fMRI as well. So how can we distinguish direct from indirect coupling? If I see a link between A and B, is it because I have a direct link or because it's going through C? Can I measure causality? Can I measure that A is going to B or B is going to A? And can I distinguish long range from short range effect using for distinguishing? So one technique that has been proposed in fMRI as well, probably by the team of Habib, is to use partial. We can use partial correlation. Here we can use partial coherence to remove the effect of another element to see if you have a true connection that exists. So that might be a bit unstable computationally, but it is interesting. And to infer causality, I'll go back to that in a minute, but it's mainly Granger causality. So here we want to see if there is a specific direction of the flow of information. Again, something where we can hope to measure something with EEG and MEG or with intracranial EEG, whereas when you apply a big nonlinear system and you show the response 5 seconds later in fMRI, this is going to be completely mixed, and it's very dangerous to look for causality in an hemodynamic signal, where here you're directly measuring. And the main one that has been used is Granger causality. So the idea of Granger causality, it's an autoregressive model. So it is the idea that you try to estimate your system X at time t from the previous measure. How can I model my system or predict my variable at time t from t-1, t-2, and so on? And the idea of Granger causality is that if you had another variable y at t-1, t-2, then you predict better. So if you have x and y past value, and if this is predicting better x than x alone, then you can say that y is probably causal or causing x. So that's one way to measure it. And we have technique to go over that. Of course, it might be a bit difficult when we have more than one condition into that, but you can use conditional Granger causality to partially out the effect. This is like the partial coherence or partial correlation. So we have partial directed coherence. We have we can measure causality in the frequency domain in specific, so partial directed coherence, or using the directed transfer functions. I won't go into the details. There are a lot of metrics, so I just want to give you a few ideas. No? So we started with coherence, which is correlation in the frequency domain, coherency with phase and amplitude, phase locking value, and now we talk about Granger to have the notion of causality. And all those models are based on autoregressive model. So parametric multivariate autoregressive model. I don't have time to go into detail, but like the order of that model is something important. Huh? How long you will go in the past to estimate your data, and so there are interesting reference here. And one of the last points I wanted to make, because this is very important, is volume conduction. So we want to understand, and that's typically a big problem in EEG and MEG, what is a true connection from what is a spurious connection. And why do we have that? So yeah, one of the first things that has been proposed is to use imaginary part of the currents, and that has been proposed by Guido Nolte. So the idea is that when you look at the currents, you have your real part. Your real part is what we call the zero lag. So you look at what's happening instantaneously, directly. And you might have zero lag correlations. You might have two neurons that are indeed acting that. But the problem, and I'll show an example in a minute, is that volume conduction. So volume conduction is how much the signal here or the signal reconstructing is influenced by the signal reconstructed here. This is only zero lag. So the idea of Nolte was to remove all that zero lag and to use only as the imaginary part. So you're only looking at phase lag. So phase lag correlation of currents makes sense, because if you imagine that you have an activity that's spread there, it requires few milliseconds to go from there to there. So you might have an activity and a few milliseconds later there. And that's exactly what we do. But we are killing the first part, which is zero lag. So what is important? Why is that important in source reconstruction in the domain of EEG and MEG? Because we have those two. Let's imagine we have this generator, and then a few milliseconds later, it goes to that generator after T2. But because of the spread, if I have two channels here, both channels will see information from this and from that. And what's happening at time T1, so what is a zero lag, is that I'm going to see at time T1, I'm going to see an activity here and here at the same time. So when the activity in T1 is occurring, I'm going to see a measure. So I'm going to see a link, a correlation, a coherence between those two channels. But this is with zero lag. And then, because the true source was correlated a few milliseconds later, I'm going to also see a connection which is lagged. And this is what you measure with the imaginary part of the coherence. But this is at the price of killing the first one. No? So we'll not detect the true zero phase coupling, and it can therefore underestimating the coupling. So, so there are interesting papers in MEG or in EEG that are showing how much it is effect, and that gives you an idea. You see a lot of connectivity papers that are based on the scalp, and the range of that, especially of EEG, is huge. So because of the volume conductions, if you have a connection, you might say, okay, I'm doing a link between an electrode there and an electrode there. They are sufficiently far away, it's okay. No, it's not. The volume conduction in EEG is very long. And it has, so it's really important to take that into account. And one interesting way to take at least partially that into account is not to do in the sensor space, but to go back in the source space using the technique I showed you at the beginning. No? So probably not the dipole technique when you are dealing with resting state, because it's too low SNR, but the distributed models, beamformer or the minimal norm or MEMs. And how do we solve that? So we have several versions of it. So either we apply a source reconstruction first, and we apply connectivity metric after. And as Abib was mentioning, there have been work on how we can tune. Perhaps the standard parameter for source reconstruction are not the best for connectivity, and they should be tuned. And it might be difficult. There are other techniques that are based on modeling technique, where you use a generated model. So the idea is you try to estimate both the connection and the activity at the same time. but usually you need an underlying model. This is usually what you have with dynamic causal modeling. So you're going to make an assumption that you have a model like that between different regions, which are linked in that way. And then you're going to use your data to infer how much this link are getting into your data. Or you can use model comparison if you have two models like that or three models to assess according to your data which model is the more reliable. But it is based on a model. If you have an underlying model like that with few regions, it works with, I don't know, but 5 to 10 regions max, I would say, because it's quite demanding. It's been done a lot by the group of Kristen for SPM, for fMRI first, and then developed by Olivier David, Jean Denisot, and many others in the world of EEG and MEG. So it's a generic Bayesian framework for inferring hidden unobserved neuronal states from the measurements, originally proposed by Christophe. It's been implemented for MEG, EEG, also intracranial LFP, and it's particularly suited to test hypotheses. So if you have a good idea that this is your underlying network, and if you want to test if this or that connection in the network got involved, that is very well appropriate. It's been used for the visual system a lot. And but if you don't have such models, that's more complicated. You have many other techniques. I will not have time to go through them. I will just show you one because Jawata showed you that a little bit this morning, which is what I showed you a lot for now was phase locking value. You have new phase metrics that are not sensitive to the zero lag, which are the phase lag index or the weighted phase lag index. These are quite interesting metrics that are removing a little bit like the imaginary part of the currents. And there are also other metrics looking at the envelope. So far we looked a lot about the phase. We can go back to the amplitude, and that's interesting. So far we looked at the phase, but if I have those two channels, I can make Hilbert. And now when we look at the model, so at the amplitude of my Hilbert, not at the instantaneous phase. And when you do that, you have a quite slow signal. Let's say this is the fluctuation in the alpha band. You have a slow signal here, you have a slow signal here. And actually what was interesting is that that signal in terms of fluctuation is not so far from the slow fluctuation of the fMRI. And you can measure correlation between those slow signals. And if you first do a seed-based connectivity map with that kind of signal, first what you get again mainly the leakage, so the zero lag effect. So you see that you have a lot of connectivity around your seed point. And that's logic again because we come from a MEG or an EEG inverse problem. But then if you remove the zero lag of those slow signals, and there is a way to do that which is called orthogonalizing those two signals, then you have the illusion that you get nothing. This would be at the same scale. But if you reuse the scale here by 5, you see that there is an underlying structure. And that structure looks very cool because it looks like a network structure. So here you have the motor complex and you start seeing a motor network. It's been first proposed by HIP. It's been nicely illustrated by Brooks I think in 2011. And in this nice study, what he did, it's a MEG study, and he compared with fMRI, resting state, using ICA in both cases. And what you can see is that with MEG, you can recover most of the resting state network that you find usually with fMRI. There have been recent studies from the group of Research and Bayer who are saying that this correspondence is not that clear, but that was a first. So it's a possible mechanism linking those two. It might not be the only one, but that doesn't change that this slow fluctuation mechanism is an interesting mechanism. And I'll show you this afternoon. We have been using that in epilepsy. And I think I will just finish with that example of phase locking value. This is something we just submitted it actually, that I did with Tamira Victor and David Poucher. So here we have all night intracranial EEG data from patients with epilepsy. So first, we removed all the channels that are epileptic. We looked only at the normal channels. And we wanted to study how is your brain reacting when you wake up. So when you wake up, some of you are waking up like that, some of you it takes more time, and we wanted to investigate that. We know how the slow oscillation are changing in the delta, theta band, and the alpha band, but we know poorly what's going on in the high frequency, probably in the gamma band, or even higher. Why? Because usually when you wake up, you move, and when you record EEG, you lost all your signal because of motion. Here we've been using intracranial EEG data. So actually, so it's data from I think 18 patients. So we have a good coverage. I forgot to remove. Actually, this is the coverage that we have here. So when we plug all the patients, so we have an interesting coverage. And of course, we cannot so we do have an interesting coverage. And what we did, so we took when the patient were waking up according to the scalp EEG, we took 15 minutes before and 15 minutes after. And then from those 15 minutes before and after, we compared, so we had it's a group study. So of course, all the patients are not impacted the same way. So we grouped a little bit like the atlas Jawata showed. So we grouped the different regions, either to look at power, just power activity, or connectivity within underlying network, and we use Yeo resting state network to do that. So we have Yeo network and the Vital Atlas, and either we look at the power before and after the waking up, like 15 minutes ago, or we look, and here, because it's a local measure in situ, we were looking at the phase, and we do have true zero-lag correlation, so we look at the PLV in that context. And then by taking some baseline data as reference data, we could measure when the data were going out, coming out, either from our sleep data, so when our data were higher than our sleep data, or smaller than some awake data, to get the time of awakening. And here you see where we measure the real time of awakening according to the SEEG data. And this is several minutes. So this is 15 minutes here, 15 minutes here. So you have like 2 or 3 minutes here. And here I show you increased activity in the irital band, so 140 Hz and above. That is occurring within those regions around that after. And here I have decreased activity in the irital band in these regions like just before when I compare to so this is when I compare to wakefulness, and this is when I compare to sleep. So this is increase compared to sleep, and this is wakefulness. So this is decrease compared to wakefulness. So it's kind of equivalent, and you have the power increase over there. And then we did the same for connectivity, so using PLV, and we found that we have an increased connectivity within many of those regions using PLV in this very high frequency band. And that's not something that would have been very difficult to get. So I will probably stop there. I have this, which is an interesting reference. So here you have 2 interesting papers, notably papers from Halva that are looking carefully what are the source of spurious activity when you're doing inverse problem. And I will just start with my last slide. So I have few general advice I was reporting. So here, just go to the conclusion. I was showing again what Jawata showed this morning, but there's no need. I think it's over time now. So in conclusion, MEG connectivity is extremely promising field of research, and the phenomena it may reveal can be fundamental from science. This is why, precisely why it is so important to use method critically and to acknowledge possible confounds when interpreting the data. We have to be aware of that. You have lots of tools, and some of the main known tools in the field of EEG and MEG, MNE, field tree, brainstorm, and a lot of other tools to measure those connections that are right now available. And I like this last quote. So this is again a slide from Karim, because the information is so rich when you have EEG and MEG data in terms of all the different metric, we have different source imaging technique, different kind of coupling metric. I did not mention phase amplitude coupling, but this is also something else. Different frequency bands. So it's really look a bit like a big, like a fishing expedition. We have so many variables. But what is important is we might catch some very big fish by doing that. And there are very important information that we might catch from that. So I think it's important. It's way richer that in terms of time domain that what we, frequency, that what we can do with fMRI, but it's more complex. And we need way more study on reproducibility, reliability, because when you look at the literature so far, you see all kinds of results, some showing increase, decrease, the source localization is different, and then the frequency is different, the metric is different. So it's really complex, but it's a very rich amount of information. And I'll stop there. I think it's way more than that. Thank you.
So thank you very much, Christophe, for this overview of the different technique of construction.